Hello guys, this will be the first of two videos. I'm going to go from slide 1 to slide around 9, number 9 during this video. You can see on this first slide here, what I've got here is your slideshow on the right, what I gave you guys, and my key here is on the left. So I'm going to go through what you guys see in your booklet and then what you should actually add to it here on the left. So you can see here that the first slide is of conjoined twins. It starts off as one cell that was fertilized, one egg that was fertilized, then a little zygote. It goes through first cleavage. It doesn't ever quite divide into two equal little turtles here. So moving on to what you guys need to be moving into. This first slide here, what I want you guys to do is, other than just reading through this and understanding what different mutations are, where mutations can be acquired or they can be inherited. Both of those are quite essential that you understand those, that some can be acquired, some can be inherited, but all mutations, whether they're acquired or inherited, are changes in DNA sequencing. Okay, so I wanted you to highlight or underline, manipulate this first statement so that it stands out. So you would go over here and put like a little box over it that kind of shades it or outlines it so it stands out. Uh, you may want to go back and also underline and or put a box around mutations can be acquired and mutations can be inherited. Okay. Whoops. Wow, that thing went crazy. All right, so for this next slide, you can see you got 3 blanks here, and this is just saying that the 3 types of mutations can be either helpful, harmful, or neutral. In other words, no effect at all. You could have a gene mutation where the bases slip over, and instead of now the expression of blue eyes, now you have hazel eyes. So is that going to be any really big harmful, detriment to your growing up or your survival? And the answer is no. That's going to be a neutral effect. It's definitely not going to be helpful, but it's not going to be harmful either. Most mutations, though, are harmful. Okay, such as this mutation here with the bison. Bison has a little albino calf. Is that going to be harmful for its survival? Well, being on a farm, probably not. But if it's in the wild, it probably will be because predators could see it far better than it could see the brown bison. Um, and so they would hunt this guy first. All right, so the next slide, I'm going to try to keep these things rolling here. This is the slide where, for whatever reason, my key here is missing the pictures. And so with the pictures here, you can see this first one is all about point mutations. And I did put this one in because it talks about the silent, nonsense, and missense and what those are. So you can go through those and kind of look at seeing what's going on with these various terms. And then you could also look at chromosomal mutations. With chromosomal mutations, it's not just messing with one base or one codon. It's messing with the entire gene or several genes all at once. So instead of having one gene that's messed up, you may have anywhere from a few to hundreds messed up. So which one is the worst? By far, it would be chromosome mutation. So over here, in this block over here, you would write chromosome mutations. Okay, so over in here, you'd write chromosome mutations. Okay, moving on to this little guy here. This thing is like really rapidly advancing. It says many kinds of gene mutations can occur, especially during DNA replication. Where is this actually being done? Well, Sometimes it's done in mitosis, never or rarely in meiosis. It's more than likely going to be in the S phase. So what I want you to write over here in a box, and I would write mitosis slash or S phase. And you would put this like in a red type and put in a box over all of this. So I would hide which is mitosis or meiosis. It's neither really. It's more S phase than anything. This is where DNA is being replicated, and when replication is not going as planned. In other words, it substitutes or inserts or deletes a base. There's going to be a problem with the DNA. But if it doesn't catch it, it doesn't look at it as a problem with the DNA. It thinks it's fine. But when it starts making proteins and it's trying to make that particular protein, that's when the problem is going to basically arise. Okay, so write down mitosis or S phase, and then basically cover this up. All right, and slide 6. Here you have that mutations are caused by several factors. And the first factor, of course, is replication. We just talked about that, and that's in S phase and or mitosis. You have mutagens, which are environmentally induced, such as you go out in the sun, you get UV rays, you go have some X-rays taken, they don't put a lead shield over you, you get exposed to X-rays. Chemicals are huge. You could be working in a lab and you didn't take the precautions you were supposed to, and you could definitely get harmed this way. So chemicals, big, huge mutagen. So the next one here is cancer drugs. So things like chemotherapy, where they're using chemo to actually radiate and kill off cancer cells. Too much, and you're going to do damage to the new cells. Too little, and you don't actually kill all the cancer cells. So it's got to be very well monitored for your survival and how well you do. Okay, so I explained Rachel Carson here. And it says that she was an ecologist who warned about the use of pesticides causing mutations in songbirds. And so why is she sitting here next to a creek? And she's in this riparian zone, which is basically where trees and foliage is next to a river. Why would she be sitting there? Okay, she's looking at songbirds, but we as humans don't traditionally go spraying pesticides in these areas. So where's the pesticide coming from? Number one place pesticide comes from is from farms. If you have a river such as this that's running through the farm and then lands up in a riparian zone such as this, the waters can still have pesticides in them from runoff. So you have a little bit of rain in the farm, the rain then runs off into the river then takes it on downstream. All the little bugs then get exposed to the pesticide, but maybe it's not enough to kill them, but it's enough to get in their bodies. And birds and fish and other things that the birds eat ingest this, and they build up this amount, this low-level amount of pesticide in their system, which then causes mutations in their own DNA. Okay. On slide 7. This slide here, we're talking about different kinds of proteins, and they are both functional enzymes. So here is functional enzymes and structural proteins. So the proteins build and proteins have function. They normally do not do both. Those that are functional are usually like your enzymes. They're the doers. And the structural ones are the builders. They build different structures, like parts of the cell membranes and what such. Okay, and the last Now, let me go one or two more. Uh, this one here, end results with mutations. Okay, so mutations may or may not affect the organism. So you can have mutations happen and it codes for an amino acid that also codes for the other amino acid that was being mutated. Does that happen? The answer is yes. Is there any change whatsoever to the protein? No. Did a mutation occur? Yes. Okay. In that case, no effect will be noticed. On the other hand, you could have a mutation happen and there's a great deal of change. So, such as a mutation may cause premature stop codon. So that would be this first set of blanks here, stop codon. So a stop codon literally means that. It means to stop. So if this is the bases, and say you have 60 bases in here, and they're from this end is the start, and all over here is stop. And you were to stick a stop now right in here, okay? So maybe 35 bases in, and then you put a stop codon. That means that new gene is only going to go from this point to this point. Everything beyond this point now is an intron, junk DNA. It gets tossed out. Is that going to code for the same protein? The answer is no. All right. An active site. So this one here, where it's talking about a mutation may change the protein shape, or the active site. The active site is the part of the enzyme that attaches to the substrate. So this part here, my right fist here, was the substrate. So we'll say this was lactose. This is the example I gave in class. So this would be lactose. And this is the enzyme over here. And this enzyme has an active site that will align itself and attach itself to the substrate. So this is lactase bonding to the substrate lactose. Once it's bound, it will change shape, which it puts strains on the bonds of the substrate and can break that sugar down now into sugars that can be digested by your body. Okay. So that's protein shape and or active site. Think of it as you have a lock in a door. If you change the lock without changing the key, is the key going to work? And the answer is no. Okay, your chances of that key working are pretty slim. And the last one here is a change in gene regulation. Again, the gene regulation is kind of like the light switch to your lights. Okay, and the example I gave during class was there are some babies that are born, they have deep blue eyes. And then like a couple days later, they have pale blue eyes. And then a couple days later, they've got green eyes. And over, like a year's time, their eyes may change to finally hazel or brown eyes. Why is that? Well, it's because these genes that regulate the gene expression are turning on and off. As they turn on and off, the eye color goes from like blue to brown. Now, does it always go to brown? No. A lot of times it will stop in somewhere along that journey. It could stop at blue, it could stop at green, it could stop at hazel, it could stop at brown. Okay. And the last slide, I want you to put in here somatic cell and gametes. So that is somatic cell, and over here is gametes. And these are in parens. Okay. Coming up on my time. So once you get that in, you're pretty well done with the first part. I will finish it off in the next video. All right, later.